And development, I'm leading the TomoAnalysis development project. So I want to show how the TomoAnalysis works and how it can be applied for your sample analysis. So today, I'm gonna mainly talk about the strategies that we use for analysis. So it's about how we identify the objects in what we observe acquired by the top HTX1 or the former HT2H. So let's begin with the characteristics of the data set that we acquire with the machine. So the sense is the data is in three-dimensional form. So we should take into account that fact. So in general, most of the analysis software in biology image nowadays, it's based on two-dimensional, but it's expanding to three-dimensional. The approach is very different from two-dimensional, so we should take into account the difference. And at the same time, holotomograph data is label-free image, so we should take into account the nature as well. So in fluorescence image, which is very typical in biological image, it's rather easier to analyze those results. But in our case, it's label-free, so we should take into account the fact. And also, at the same time, the instrument can also acquire fluorescence image at the same time. So we should combine the two, which is HT and fluorescence, to get richer analysis about the data set. And also, we acquire the data in time lapse. So we should take into account tracking nature of the samples. And by tracking the object in time, we can get more abundant data from the measurement. And as I said, the data can be acquired from HTX1, which is the recent one, as well as the former one, HT1H or 2H. So we take into account the compatibility in data analysis. So that's the basic characteristics that the TomoAnalysis should handle, and we wanted to focus on those features. So let's begin with how we choose the analysis software. So when we use a commercial microscope, the basic approach is to use the proprietary or the software that is supplied by the manufacturer. So it's very easy. So the work process or the analysis protocol is well defined, and you just follow the streamline. That's because the image format is dedicated for a certain type of manufacturer or instrument. So there's no much deviation possible. But in our case, it's a bit different. So holotomography is quite a novel technique. So you need to, or you want to apply a new analysis approach to get rich information from it. In that case, we use open source software. Open source software, it's at the beginning, it's very cheap because it's most of the time it's free of charge. So you can get anything in the website and download it to try the analysis. That's a small thing. The most important part of open source is that it can be flexible because many people contribute, and so the structure should be very flexible to accommodate all the variations. And at the same time, because many people contribute, there are very novel approaches there. So you can select very new or very intriguing techniques to apply those techniques to get more information. So that's the reason why people are so open to those open source software. And at the end of the day, when you have very large set of data, you want a more efficient way to analyze those data. Then you use a programming language like C or Python to have your own script to run any specific target very efficiently. So we want to incorporate those features. So TomoAnalysis, it's for our own format TCF or holotomographic data. And we accommodate those open structures to accommodate new approaches or interesting approaches. And at the same time, we develop the code to optimize the performance, to have a better performance on analysis. So let's see what is the basic approaches or the strategies that TomoAnalysis can do. So this is the basic flow for bioimage analysis. In the center, you see this segmentation. Segmentation means that you identify your target from the background. So this is the most important part. And this is all of the analysis at most of the time. And for this segmentation, you do some kind of pre-processing or post-processing when you need an extra job to refine the segmentation results. And when you get nice segmentation, you do quantification analysis based on that segmentation results. And TomoAnalysis follows the same flow for the analysis. So the most important part here is the segmentation. So we can divide or classify the segmentation approach in three. Actually, it depends on the nature of the experiment. And at the same time, it can be classified in terms of difficulty. So the first one is when the sample or the data has fluorescence channels. Fluorescence channels means that you stained or you label some targets. So you can identify the target by the fluorescence signal. And in most cases, fluorescence signal can be identified by just simple thresholding. And for that thresholding, you do some kind of pre-processing or post-processing to reduce noise or to refine the data based on the results. So most of the cases, fluorescence text objects are very easy to identify, not only for TomoAnalysis, but for most of the softwares. And the second part is when the target has uniform structures, like in our case, lipid drumlet or mitochondria. In that case, the structure can be defined very simple or a combination of simple O's. And we take advantage of that simplicity in refining or in identifying those uniform objects. So the strategy is quite similar to the technique that we use for fluorescence tech objects. In this case, just simple thresholding may acquire the uniform object like LD, and sometimes 2 or 3 combined methodologies can help to refine those objects. And for those cases, we use simple rules to acquire those things. And the example is like lipid drumlets and mitochondria, as I've mentioned earlier. And the last part is when the target object has very complex structures or internal structures, so like cells or nucleus. In this case, because of the complexity, it's very context dependent when we want to segmentation, when we want to segment those objects. So that means it is very difficult, and it is very difficult to generalize the segmentation approach for those internal structured objects. So example is cells or nucleus and other textures like in tissues. So we divided the strategies according to these 3 classifications. And I'll show the difference in the following pipeline presentation slides. So this is a summarized image of the 3 different segmentation strategies. On the left side, it's actually stained nucleus, or nucleoli. So in that, on the left image, we segmented by thresholding to identify the fluorescence type nucleoli. And from this segmentation, we got these analysis about these targets. And in the middle, it's uniform example of uniform object, which is lipid drumlets. And here you see very colored dots here, which is individually segmented lipid drumlets. And the quantified measurement value are listed in below. On the right side, it's the example of object with an internal structure, which is cells in this example. So each cell is identified, and its volume or dryness or other physical quantities are analyzed as shown below. So this is the three typical examples of the segmentation strategies that we have. So let's talk about the strategies again. So these four steps are the basic steps for segmentation of bioimage analysis. And for these to establish or to proceed these strategies, we applied a modularized approach to do this. So it means that we can apply the strategies for various targets by by combining very diverse methodologies. And once we get the strategies for identifying those targets like LD or cells, then we can combine those strategies to have a bigger or more advanced or complex analysis. So we can segment multiple targets at the same time. So for example, when there is large LDs and small LDs, then we can do it in a divide and conquer way. So we segment large LDs and small LDs separately and combine the two results to have better understanding about the LD distribution. And also we can do some kind of relational analysis, which means we do the LD segmentation and cell segmentation in a separate way and mix the two or combine the two to have a cell-based analysis, which means the amount of LDs for each cells like that. So we can make a quantified analysis on the cell basis, on cell basis. So this is examples of combined analysis. So on the left side, we stained two different organelles here. One is nucleus and the other is nucleoli. And we do the segmentation separately and combine the results. And on the right side, we can compare the difference in the density or the RI value of each nucleoli groups in each nucleus. So it shows a bit of differences. So like this, we can get a combined analysis. And the second part is some kind of interesting approach. So in this case, we stained drugs and treated cells with drugs, which is the drug is and the drug is tagged with fluorescence. So in this case, when we quantify the amount of drugs, so we can identify the amount of drugs in cells by segmentation based on fluorescence thresholding. So on this image, you can see the fluorescence signals inside the cells. And because we can segment those cells individually, we can quantify the amount of drugs intake inside each cell based on these segmentation results. So from this, you can see the amount of drugs per each cell, which is listed here. So in this way, by mixing the two analysis, we can get more advanced and more sophisticated analysis. So the reason why this kind of approach is possible is that we build the analysis approach by combining many processes step by step. So by mixing those processes, we can do what we want as we imagine. So there are processes for pre-processing, and there are segmentation processes like this, and post-processing process like this, and quantification processes like this. So by mixing those processes, we can build what we want. So by mixing those processes, at the moment, we have 6 pipelines, which is validated in our company. So here you can see number 1 and number 1-1 is fluorescence-based measurement, which acquires the fluorescence amount based on thresholding. And the number 2-1 is lipid draw analysis. And number 3 is instant segmentation for cells. So this is the basic strategies to identify those simple targets like fluorescence, LD, or cells. And by mixing those four segmentation strategies, we can get these kind of advanced strategies. So 5-1 is fluorescence intensity of individual cells, which I've shown you in the previous slides, which takes the amount of drugs inside cells. And 6 is lipid drum analysis based on cells. So we can quantify the amount of LDs per cells by combining the two approaches of LD and cell segmentation. And the quantification parameters for measurement are like this. So we measure the quantity in 3 dimensions. So we have geometric parameters and physical parameters like this. So in geometric parameters, the centroid coordinate in XYZ, and also center of mass coordinate in XYZ, which means the data is in three dimensions. And also we have physical parameters like dry mass, concentration, and Ri mean, max, minimum, and range values. So after we get the segmentation results, we can make it visualize like this. So based on the analysis, we can do more nicer representation of the results, which gives a better impact on when we want to present the results. And now I'll show how the TomoAnalysis works in real situation. So this is the basic analysis workflow for TomoAnalysis. So the strategy is the main engine of the TomoAnalysis, and to use those engine, we need a kind of platform or the workflow. So the first 3 section is for preparing the data. So first, we load the data, and second, we select channels and time points for our specific analysis. So when we have time-lapse data, we can specify a certain time point for our analysis, or we can select all the time point for our analysis. And optionally, we can make a region of interest for our analysis to minimize the other unwanted signals from the analysis. And after preparing the data, number 4 and 5 is for the real analysis. So at step 4, we adjust parameters for a certain analysis. And when the parameters are set, then we run in batch to get the analysis in a repeatable or reproducible manner. And then at step 6, which is the last one, we review the real analysis results to see if the analysis results is fine, or we need to run it again with different parameter set or different approach. So that's the basic workflow for TomoAnalysis, and that is what we do for the TomoAnalysis. And let's see what happens when we segment those fluorescence signals. So on the very left side, it's the fluorescence signal overlaid on the hollow tomograph image, and which is the cell stained with nucleolide staining. So here on the left side, you see bright green spots here, and by segmenting or using a threshold value of certain number, you can get these thresholding results, and you get these analysis results. And on the right side, this is the pipelines that we use for the analysis. So in this case, it says it consists of 5 steps. So number 1 is for pre-processing. Number 2, which is thresholding, which is the segmentation step. Number 3 is post-processing to refine the result. And then number 4 and 5 is quantification steps. So in this segmentation pipeline, the most important part is number 2, which is threshold. So by defining the number, here I used 250, we can segment those results. And if you are not satisfied with the result, then you can adjust the numbers here. And by adjusting the numbers, you can achieve what you want. And as I said, the fluorescence tagged labels are relatively easy to do and to understand. And that's what most of the biologists are familiar with. So I showed these examples at the beginning. And this is the example how the TomoAnalysis works. in terms of fluorescence. Like this, it's very short time. So I skip, I reload one by one. So this is the data we loaded. And this is the segmentation results. Here, I intentionally set the threshold value very high. So you see there are many, many holes here. And we want to fill these holes. And because there are so much holes, I don't think it is filled with these particular objects would not be filled. And yeah, yes, this is the post-processing result. So these parts are filled, the holes are filled, and these are not filled enough, which means that we should adjust the threshold value to have a nicer closing of the object. So this is I showed this one because to show the example of adjusting threshold. So after getting this refinement, we do the analysis. And to identify each object from one another, we label the object with different colors here. And the object analysis based on these objects like this. So this is the simple example of fluorescence type objects. And this is the zoom in view of the fluorescence, the pipelines that I used. So as I showed, it is very short and it is very straightforward. So you can be familiar with this kind of strategies. And the second approach is the uniform objects. And in this case, it's still relatively simple, but it's more complex than the previous fluorescence, the strategies for fluorescence. And here, these results could acquire by using these two different threshold criteria. One is absolute value of Ri, which defines that if the Ri value is higher than this number, then it can be a candidate for LDs. And the second criteria is relative brightness or relative Ri value changes compared to its neighborhood. So in this case, if the value is brighter than by this amount, then it is considered the LD. Here, these numbers should be divided by 10,000 to be translated into the value of Ri. So it means that the spots should be brighter than by 0.018 in terms of the Ri value. So by combining the two criteria, we can get these kind of objects. So this is the result for each criteria. When we use Ri values higher than this one, we see most of the LDs are identified. But at the same time, we also got these kind of areas as well, which is the condensed membrane. So when the material is condensed, the Ri value gets higher, which means that Ri is not that specific in terms of material distinguished materials. So we use the second criteria here. So the relative value brighter than 0.018. In this case, it's not included. So by combining the two, or by taking the intersection of the two objects, we could get the object of the LD analysis, like I showed before. And this is the screenshot for the LD analysis. The LDs are so small in this case, so it's not that easy to identify. So what you've seen here is that as the analysis process goes on, and each process produces its own output in the middle, then the data is shown or stacked in these layer controls. So you can see many outputs in the middle. And by these outputs, you can tell which processes was enough satisfied or acceptable for the analysis. And by combining those results, you can see the result. And also, you could see that the segmentation result is applied to the visualization to see in three-dimensional representation. In that way, you can get a more idea about what you get from the analysis.
Sorry. Can we have a question? Because what can these act as a question about the thresholding method? So is only manual thresholding available, or can we use other methods like segmentation?
Oh, that's a very good question. At the moment, we mainly focused on the pipelines or the workflow. So at the moment, it's all manual, but we will incorporate those automatic thresholding method. There are a bunch of the automatic parameter adjustment method, and yeah, we want to have better understanding which one is optimal for the pipelines. So we will provide automatic thresholding method as well, but it's not in the software at the moment, and we will release this soon, and we will let you know when we have those features.
Is pipeline available?
Sorry?
Pipeline available right now in the software?
Yes, there is.
And can we stop in between the pipeline and do some manual filling of the whole, or choose some manual areas for segmentation? Like, is there an option like we can edit one step manually? I mean,
yes, that's very, very good request. Actually, it's not here at the moment, but we are developing that feature as well. So it'll be available at the end of this month, actually. So you will see the feature soon.
I have one question.
For
the liquid droplet, you mentioned two conditions.
That's right.
RI threshold.
Yes, which one is absolute and the other is relative?
The delta RI.
Yes, a relative one.
When you say delta RI, it is related to the neighbor.
Yes, right, neighbor.
Pixel.
That's right.
So then in that case, you will only have, let's say, like the boundary labeled. If the limit is uniform, let's say 1.37, and it's a uniform circle of, let's say, 1.37, then it is the boundary that is only getting segmented, not the inside.
Right. Yeah, for that I made you a bit confused. So here, the meaning of neighborhood is not the nearest neighbor. You can define the area of neighborhood. So you can extend the radius of neighborhood to define the region or to define the size of the LDs that you want to focus.
Okay.
So if there's big LDs, then you can set the neighborhood larger than that big radius.
Yeah, but then the problem would be some of the nucleolus also might, because their area is big, so kind of a trade-off there. That's
right. But very interestingly, yeah, those nucleus, the value of the absolute RI value does not coincide with the criteria. So we can exclude in the other criteria.
Okay.
Yeah. So that's the reason why we combine the two criteria.
Okay.
So as you said, there can be many different situations. So pipeline supply is very flexible, and if you want, you can edit your own pipeline based on our approach. So that's the reason why we use these kind of flexible structures. So this is another example of uniform object segmentation, which is mitochondria. So on the left side, you can see a very vague pattern of mitochondria. And on the right side, you can see the mitochondria segmented based on that patterns of the mitochondria. So here, it's not perfect one. So I did not show the pipelines or the the screenshots where I show the examples. So here you can see some defects like this. Here it's on the membrane, its membrane, and it's mistakenly considered or segmented as mitochondria. So we should exclude those one as well, but not at the moment. So I show this one as an example for those strategies for mitochondrial segmentation. And the last one is segmentation of object with internal structure, which is very tough and complex to understand. So here, this one is the simplest form of the segmentation strategies, which is for cell segmentation. Here, the strategy is to find the center of the cells from high RI values, and then lowering the RI value to expand the boundary of the seed to the boundary of the real, the boundary of the cells. So on the first image, we identify the cell center, which is mathematically calculated. And then we lower the RI value for growing the cell size like this. So successively, we extend the cell surface to the very boundary of the cell and reach the final boundary. And from this approach, we can segment those cells even though they are touching each other. So in this case, we use watershed inside the process, but the basic watershed approach, which takes the boundaries and then making the boundaries separated by the simple watershed algorithm doesn't work in this case because it's three-dimensional, and watershed is the only way doing the segmentation based on the boundary. But if we do not make this kind of divided approach, then most of the cases simple watershed did not work. So this is how the cell segmentation works in our software. So you see these series of processes here and the outputs in the middle like this. So there are so many outputs in the middle. And if you get that segmentation, you can do this kind of 3D representation. And so because our data is in 3D, you can use those 3D segmentation results to get a nice visualization like this. And also because it's individually identified, we can highlight one of them as we want. And we have a diverse strategies by combining the strategies that we find. So for example, we do LD or mitochondria segmentation and make them quantified in terms of per cell measurements. Like, so by using HT, we can do this kind of analysis. And also when we have fluorescence tech signals like drugs, then we can quantify the amount by cells, like I showed before. And also when there are multiple channels of fluorescence, we can analyze the data using both the fluorescence signals, as I shown before, like the nucleus and nucleoli. So some people will ask, this is the frequent questions that I got. So does TA handles data from the former instrument, like 1H or 2H? So yeah, the answer is yes. The process in TA is compatible with any data type that is included in TCF. So you can analyze those data from the 2H instrument as well. And the second question is that, am I able to open the analysis results in other programs like ImageJ? So you have seen the outputs in the middle are listed on the right side. All the data can be exported in TIFF or CSV format to be used in other programs like ImageJ or other analysis programs. So you can do your own analysis based on the analysis the TomoAnalysis provides. And the third question is, does TA support machine learning or deep learning algorithm? So this question is I got because we supplied the former analysis software with AI enabled analysis features. So at the moment, we do not provide the feature because the structure is not prepared for accommodating those machine learning systems. But we are developing the structures for accommodating AI models, including other well-known models like elastic or units as well. So soon you will have the features to use external models for AI as well as the AI models that we provide. So you will have the features soon. So yeah, so the thing is that it will provide functions to use other programs like elastic or units in headless mode. So you can take advantage of other open source approaches or models to have AI-backed analysis and combine the result with TomoAnalysis. So that's the thing that I've prepared for this time. And if there are any questions, please ask me.
Software updated now. The current one we use has that AI segmenter that you mentioned. So we can still use that AI segmenter, or will it be updated?
So the model, it's the AI model, the model that we have for our old software is in version 1.3. And when we provide the new software, it will not, it will be replaced. And if you want to use the former version, then you need to install in a separate folder. And you can use both of the software at the same time by installing in other directory. And as I said, we wanted to incorporate the AI model in the new software. So in maybe early next year, you will have the function that runs both the AI models of the older software in the new software, as well as the features that we have I have just shown you.
Tissue model also applied with this HT. For the segmentation, tissue is some is up, some is down. So do you do also 3D segmentation for those things?
So yes, that's very, very nice topics that TomoAnalysis want to address. And at the moment, what we find is that it's very difficult to segment cells because it's very confluent. It's compact structures, tissue. But what we found is that we could extract the morphological aspects of the tissues. So based on that feature, we can separate some of the specifics of tissues. I don't mean it can segment those cells individually, but we can extract the characteristics of the tissue by with a certain resolution of spatial resolution. So yeah, I think we can do something in tissue analysis as well.
Visual fluorescence microscope has the different resolution for X, Y resolution and G resolution.
That's right.
So Tomocube, how about Tomocube? Is it all?
Actually, it's a fate of all optical microscopes. So yeah, the resolution in XY is much better than the resolution in the G direction. So that's also applied to the data acquired by Tomocube systems. And
Can you
in
just briefly explain how the optically collecting the image is it similar to conventional microscope? You collecting the refractivity.
right. So the optical configuration is the same as usual transmission light microscope, but how we acquire the refractive index is very different from most of the optical microscope. So it's like so in a nutshell, it takes like it's like G stacking, but for every Z position, we take multiple images with different light profile. So by using a different profiles, we can simulate the light beam, the angle of the light illumination. So in principle, in tomoholography, the light angle, incidence angle changes in 360 degrees. So it rotates. But in this new system, it does not rotate. Instead, it simulates the rotation by modifying the illumination pattern. And for that pattern, for the same pattern, we do G stacks in G direction for like 60 microns. And by accumulating and by comparing those G stacked images, we can compute the RI distribution in 3D. Yes, that's the basic idea. So here the difference is that most optical microscope, they for each pixel, they get the direct information from the optical measurement. In our case, from the direct measurements, we recompute the RI values for that particular voxels. So that's the most different part.
We have the two questions.
Mm-hmm.
And you should turn off. So the question is about the 3D visualization.
Mm-hmm.
Turn off the speaker on your laptop.
Okay.
Yeah, so the okay, I'm just read the questions and then I will answer the answers. So the first question is about the 3D visualization. So is there any way to export the 3D cells rendered in the TomoAnalysis into any other 3D file format such as the PLY or the object, the OBJ?
Actually, I'm not familiar with PLY or something like that, but the output of the labels it can be exported in T format, and the T format itself is three-dimensional information. So if anyone can import those T in any analysis software, then they can visualize using those 3D information.
Okay, so there is another question. So is it possible to use other geometric parameters such as sparsity, volume, etc., to further threshold or select just object for the further downstream analysis in the pipeline?
Oh yeah, sure. So there is another filter or post-processing filter, which takes the measurement values as a second filtering criteria. So, but from the measurement results, you can refine the objects in a more narrow manner, in refined formats as well.
So, in your backend, do you take one slice and see if it's a circle or not, or do you take a whole sphere as a whole? Like for lipid droplet, in your backend, you select the center and see if it's a sphere, or you go slice by slice and see if all the slices are circular thing or not.
So yeah, that's very fundamental question, because in our case we take Z slices, and because the resolution in X, Y, and Z is different, the LD the shape is that spherical. So in any slice it looks circular, but when it is combined, it's not sphere, it's more like rugby ball. So yeah, it depends on the sample and the size of the LD, but yeah, you should be careful about interpreting the results. But at any rate, in any slice, it is circular anyway.
And then the lipid volume, for example, that is displayed in the analysis result.
Yes.
So that is also like approximation of the whole rugby shaped volume, or it's simply the sphere formula calculating the radius.
Oh, it's a real number. It's a real number of voxels. So it's the size is the size of the lipid is calculated from the volume. So it's circuit which ESD, which is equivalent sphere diameter. So the number is given by that in that way.
Thank you.
Okay, so I guess that there is no further questions. Then we can have some break, and then if you can show some hands up.
Yes.
That would be great. What do you think about it?
Okay, let's take 10 minutes break and then resume with the real hands-on operation of the analysis software to see how that how it goes in real. Yeah, so yeah, take 10 minutes break and
but there is another question. So after answering this question, then we're gonna have short break, and then from 11, then we will continue some hands-on the session with the same webinar. So just the last question is about the voxels. So the voxels are assumed to be isotropic, although the actual measurements are not.
So actually, it's not isotropic because the size of the voxel in XY direction is about 150 nanometer, while the Z direction, the length is about 1 micrometer or less than that. So it's not that isotropic. So we should take into account the anisotropic manner of the voxel. So yeah, that's quite tricky to interpret actually. Okay, so let's take a 10-minute break.
Okay, so thank you for attending the first session, and then we will continue from 11.
Okay, thank you. During the process of reconstruction, it involves deconvolution and schema. Even though we take the points per function into account, there is basic information. So even though we apply to any technique that we want, still there are some basic information that navigate the rotation in zero direction happens. When you see the data, you can see many blurry or something like that in the data. That shows up because of that reason.
This technique you don't need to worry about.
So when we are confident there is very weak absorption when the line is the value. For example, however, for and it's very bright. It's as if there are very high values, and when there is absorption, then it is as if there is a blank, which is very blue or
So, for the hands-on, you need to use this setup, right? Yeah, so that I should change it. I think that, yeah, from the hands-on, you don't need to stand on. Understand that you can just move on this table and then that's all like that. But we ordered some pizza, so it will be arriving in an hour, just before 12, I guess. And then, so let's just have kind of the free discussions and then the Q&A sessions would be great.
Okay, so as the second part, I want to show how the TomoAnalysis works in real life. So this is the remote screen that I use for the analysis. And you can see the outlines of the screens. So briefly, if I explain the UI here, on the left side, it's the pipeline and how it works and what's the outputs or inputs it uses. Is that the information is listed here. On its bottom side is the file. Yeah, on the bottom side, it's the files that we want to analyze. So if we want to add more files, you can use these panels to add more, or you can remove from the files here. And on its right side, it's the parameters for each process. So you can select any of the parameters, and its parameters are shown here. So at this stage, the process is, if you want to control any process, any parameter, you should visit each process to adjust. So at this moment, it's not that friendly for users, but it's for the flexibility. So bear with this at the moment. And on the middle, that's the images that we are dealing with. So we see many information from here, and you can switch on and off the data sets as you want. So here it's so here you can see the 2D slice of the data. So here I showed the HT overlaid with the fluorescence signals, which is actually drugs. So you can see the drugs are inside the cells. And there are many outputs in the middle. So you can on and off to see how it goes. So if I explain the outputs here, the fluorescence signal, we apply the thresholding segmentation, and the result is like this. And we do some kind of filtering here, but it did not change much because of the nature of the data, the sample. And based on this, we do some identification of the object here to make it quantified. And the result is like this. And all the things you can run on, you can control the process on the left side. So here, when you press this single triangle button, it runs the pipeline once to test a selected data sets. And when you press the double triangle here, it uses the parameter to analyze all the files listed here. So let's try one of the example. So I make this data set active, and you can see a very vague signal here. I'll change the value here to make it visualized. And here we see bright depth signals here. And let's just run this small single triangle to see how it goes. So it gets the segmentation, post-processing to fill the holes and identify objects. And from the objects, it gets the result like this. So the reason why the process takes a bit of time is that the data is three-dimensional, so it takes some time. So it's compared to the 2D analysis, it's just a single image, but so it's quite fast. But in our case, it's three-dimensional, so it takes a little more time to do the job. So please have patience when you run the analysis. So this is the basic idea of how the thresholding or the fluorescence tagged object thresholding. So it's quite straightforward. As I shown you before, it's done this in 5 steps. Here, but what I want to emphasize again is that you can see all the data in the middle to see how it unfolds. So let's see again. So at the beginning, it was like this. No. Where is it? Oh, yes. It was like this. And we do some kind of smoothing to have a better analysis. So we do some kind of pre-processing to have a nicer thresholding. And then we do the mask based on this one. So this is the basic thresholding. So we see many holes inside the nucleus. So we want to fill those holes. So we do post-processing to fill the holes. So it comes like this. So on the border, you see it's not filled because it meets the border. And this is the result of the post-processing, which is the identification of the fluorescence tagged area. And then when we want to quantify, we identify each object like this to have a separate measurement on each object. And then the result is like this. And a good thing is here is that we highlight each object like this. So by clicking the object, you can see which one is which and take the measurement. Okay. So you can see the volumes or the mean intensity or other physical properties. So, and also you can click this area to find a certain object of of a certain rule. So in this way, you can find the target that you want to analyze as well. Mm-hmm. That's right. That's right. That's right. That's right. And also it glitches the Actually, in that for that one, we don't have any good starting points because it depends on the sample very much. So it should be your experience about how to control these spacing for the fluorescence measurements. So suppose, assuming that you are familiar with the G setting, the analysis software takes into account the different spacing of the HT and fluorescence. So it automatically harmonizes the G spacings to get a best analysis results. So it takes into account the different spacings. And from these results, you can do the 3D measurement. So here, you choose the data set and you choose the label and make it visualized, which comes like this. Come on. Oh yeah. And my screen is very dark, but you can see the image will rotate. So I would like to change the contrast here, yet it's not automatically controlled. Oh, too bright. So here, I use the mask, identify the thresholding, and apply the mask on the HT data. So you can see here the nucleus only, which is identify the fluorescence. And you can overlap these nucleus images onto the background as well. If you modify the pipeline, in this case, it's not modified, so you only see the nucleus here, but it's possible. And here you can blend the colors following the labels identified for each object. So from this, you can see how the fluorescence distributes in 3D. So that's how this analysis and visualization works in combination. So this is the basic workflow for the analysis. For the workflow explanation, I used this fluorescence pipeline because it's very simple to understand. And let's get to the LD. It's like this. I already run the sample here, but I would like to choose another one. Make these particular workset active. And you can see bright dots, which is LD. And this consists of 7 processes, and the thresholding parts are number 2 and number 3. So let's run the pipeline to see how it detects. And in the middle, you can see the triangle marks here, which shows that the data is not available yet. And as it goes, the triangle disappears and the data shows up here. And you can see the red dots here, which means that the process identifies some parts based on the criteria. And by mixing the two, we can get this kind of analysis. And finally, the analysis results shows up. And as I shown before, the results can be visualized in three dimensions as well. And here, as I show, the most important part is number 2 and number 3, which defines the threshold values. And by modifying these numbers, you can adjust the sensitivity of LD detection. But as far as what we found, the value is almost constant regardless of the sample. So I think we should, you can use the pipeline as it is in most of the cases. And the result is listed like this. And if you press this double triangle button, then it runs in batch. So actually, I run this before, so it asks if you want to proceed the analysis again, then again. Then it goes from the first one to the last one by one, and proceeds the pipeline to get the analysis done. So after finishing the first data set, I would like to abort in the middle to see the results. So let's abort. You can abort and resume afterwards. And it takes some time to finish the process. So we know that the first one, the analysis is complete for the first one. So if we want to review the results, you make the dataset active again and reload the files, the result from here. And you can see the data is reloaded like this. And you can see the results. So you see some of the parts are not labeled here, which means that even though it looks like LD, and we are sure that it would be, but it did not meet the criteria we set. And if you go on other G slides, you see that the LD is labeled. So it means that the data set or the data is in 3D, and in human's eyes, sometimes it looks like LD, and it's on your discretion to include those parts or not. But the basic idea here is following the basic criteria, which is more like general. And it sometimes in human eyes, you think you need more modification to track all the things, but just be cautious because the data in 3D, and you should go up and down like this to see if the criteria really missing or not. So in this way, you see how the LD is detected here. Yes. Even the section that it doesn't show. That's right. That's right. So in this way, you can see that the analysis in 3D. And the last part is about cell segmentation. And load the data. Oh, it's not the data that I want. It's not. Maybe. Okay. Cell instance. Yes, this one. Add more, like this. So here I want to show how the data is selected. So in holotomography, the data is, you usually take time lapse, and you want to define a certain time point, or you want to analyze all the time point. So you can select the time point that you want from this file list. So here you can see this chain. So when you press this button, it's quite complex, actually. The reason why is that we wanted to select the very time point that we want to measure. So you'll see how it is done here. So in this case, the data contains in total 50 time points. So it's a series of 50 consecutive TCF files. And because it has 50 images, it says the start is 1 and the end is 50. And if you choose interval 1, then you can select from 1 to 50, which is 50. But most of the time, you wanted to make it more brief. So let's say you want to take interval 10, and then you can get this number, 1, 11, 21, 31, 41. And you want the last one included, for example. Then you can add this one like this. So you can manually select the files like this. The benefit of having these kind of complex user interface is that you can edit as you want. So for example, here, if the 21st images is corrupted with debris like that, then you wanted to use a neighbor or its neighbor frames, for example. So then in that case, you can edit it like this, 20 instead. So by this way, you can make the analysis more complete. And you can say apply, then it is like this, and select OK. By this way, you can select the certain data sets that you want to use. And for the cell segmentation, yes, let's select one of the data sets like this. So you can see the data looks like here. And for your information, makes it really visualized. So data looks like here. It looks like this. And yes, we don't know if the pipeline is well configured to segment those cells, but let's give it a try with the default value. So it processes, and so you see the boundary expands. And because it's very complex, it takes some time. So just bear with this. The size physical or the data size? This one, I believe each 10 points requires 160 megabytes space. So you can see the CS rows to have the cells identified. And the only big fault here I see is this one, which is tightly bound each other. So I think most of the things are done. And let's make the transparency control so you can see the cells are nicely segmented, except this one. And if you want to separate this one, there will be two ways to achieve that goal. First one is to change the parameters here. And I don't think it's worth, because it's just one object like this. The other strategy that we can do is using manual editing, which is not yet available, but we will soon incorporate the feature in the software to do that. If that feature is in the software, you can load this label mask image into the editor, and you can divide the cells by manually and use the edited mask to get the result again. In that way, you can better or refine the results. And when you get this kind of segmentation, you do the visualization again based on the labels that you made. So before, it was like this. Come on. So on side, you may identify some very vague patterns here, which is the missing information that tomography loses. And in this case, because of the segmentation, it's clearer than the previous one. And with this, you can have a nicer identification of the cells in 3D using the information that you acquired during the analysis. So you can use this one without any analysis. And if you have some analysis result, then you can do this kind of visualization to have a clearer representation of your data. So this is the basic idea of the cell segmentation, and it's all in 3D. So as you see, you can browse in up and down to see how the segmentation result is changes according to this direction. And you do the same thing in batch by pressing this double triangle button to process all the data, and you can get the result in TIFF for the image data or the mask data, and the result data is saved in CSV, which can be loaded in the pipeline part. So if you open the file, you see this export folder here, and the data saved in by the process names. So HT means that the result is saved like this, and this is the mask. And this is the measured results, which is in CSV file. And the thumbnails. So when you run the sample, the images shown in the screen is saved at the same time for your review. as a simple representation of the result. So here it's not a real data. It's just image of the screenshot. So it's for your own reference for the analysis. That's how TomoAnalysis works in batch. And I believe someone asked in the Q&A session that will the cell instance ID be conserved when performing segmentation across time points? Yes, that's the portion of tracking. So at this moment, the tracking is not available. So you should do it manually at the moment. And the tracking is quite tricky because, you know, the cells divide and sometimes dies or goes away out of focus. So we should take into account all the things together. And that's quite tedious job to make it perfect. So that's why we are not focusing the tracking feature at the moment. So bear with us for that. And I believe another person asked about the parameters. So when you changes the parameters and run the analysis in batch, the parameter sets are saved at the same time. So when you review the result, you can also see the values used for that analysis. So you don't need to write down all the parameters during your experiment. Rather, you can see or you can reload the parameter set that is used for a certain job or batch test. So that's incorporated in the analysis pipeline. So I believe those three pipelines are the basic pipelines that we can use at the moment. And the other pipelines are a combination of these approaches. So when you want to see or understand the pipelines, then for that, just use these pipelines, these three basic pipelines for your understanding, and you can easily understand the other pipelines because it's the combination of the two or more than that. And I'd like to get some questions if there are.
Well, actually, in your gave some questions online. So the first question is will the cell instance or the IP be conserved when performing segmentation across time points?
Oh, that's the question that I just answered. So it's about the tracking. So as I said, we are not focusing on the tracking function at the moment because of the difficulties that I've mentioned before. So you should, if you want to do tracking, then you should do by yourself at the moment. And it can be done you can use the exported data sets to get the jobs done. So there are areas for that absence.
The exported data sets is the color or the number mentioned, like in the current view, if I want to see the data row one corresponds to which cell, then I have to check with the color, like the red or so I mean
go ahead.
In that data row, is there any mention of what is the color or what is the number that which cell
So is the question that if the labels here is related with the images here by any like color. So here, when you press the highlight button, the value here is linked with this color. So let's say green one, you can see this green one is highlighted. So in this way, you can identify which row corresponds to which object here. And if you want to
open it in the software, I can't export the data set separately and in the software only I have to highlight it in this step.
So, So for that, the export label has numbers for each object. So if you look into the label image, then you may understand what the label data means. So you can easily identify the object by numbers. So if you import the data in, for example, image J, there is a number, and you can identify those objects by the colors or any kind of method.
Okay, then if there's no further question, then we can
Okay.
wrap up the today's webinar. Yeah, like that. So if you have any other further questions, then please just reach out to me. It's on email, sld@tomocube.com, and then we'll also send you the follow-up emails about the sharing the video record today, and then some kind of arranging a further one-on-one meeting. For the further discussing about your research proposal, and then preparing the TomoAnalysis, the one-on-one hands-on demo. So thank you all today, and then have a great rest of the day today. Yeah, thank you.
Thank you very much.
Thank you.
There's no difference. We provide it as a so. You need some kind of license to activate the software, but we at the moment supply it with 1 year free demo license, so you can try the software if you ask us.